The opportunity to represent my colleagues and present the now long-term data from the JULIET trial, which was a drug, Tisagenlecleucel, which is the modified autologous T cells from a donor against patients who are suffering with relapsed refractory large cell lymphoma. The trial is continuing to accrue to its target, but this is the data we have now, I believe, is presenting for about 100 patients that have been treated on this phase 2 trial. And I think the novelty of this trial is we're in this time point is that we can now look at the outcome of patients with relapsed refractory large cell lymphoma when compared to the natural history of what we would have expected as clinical oncologists and clinical hematologists. These are patients that have a very forlorn prognosis. You know, when we take a look at both historical data by either prospective trials or retrospective trials, we would have predicted a 15 to 20% survival at 2 years. We would have predicted a very small percent of patients will be in complete remission, you know, if that'll be sustained at that time frame. And that was the target. Most of these patients weren't candidate for transplantation, or they had a prior transplantation that failed. Over 50% of them had a prior transplantation. And to the point that we would anticipate that most of them would have had be offered a ongoing salvage chemotherapy as palliation without really an attempt at or expectation that there'd be any meaningful survival. This study, I think there's several studies that have been looked at a similar population, and I think all the studies have been, you know, have changed the landscape of how we're going to treat these patients going forward. I think what we see is approximately 40% of the patients are now alive and well, but also significantly, a lot of the patients are having responses that are durable, and a response is either where we with imaging, with radiologic imaging, we're looking to see is the disease gone, or is it reduced in size. One of the unique aspects of the study is you had to have had active disease. If we had a patient who had multiple relapses and like their third attempt at therapy was able to get them to a complete remission, they couldn't go on the trial. They had to have visible, detectable disease, so you know that they're if you don't do something, the disease is going to continue to grow and lead to their death. You know, that was part of the trial design. It allows then to measure do you actually get a biologic benefit from the cells that you're infusing. When we did that, again, we have a 54% response rate. You know, 40% were complete remissions, 14% were partials, which is a reduction in volume in 3 dimensions of about approximately 75% reduction in volume, but not 100%. And I think what was interesting and important to this therapy, as well as these whole issue of immune therapies, are that as opposed to when you give a chemotherapy or give an antibody, you get your short-term response. If you don't do something else, the disease is going to start regrowing. Here, the cells are viable, and they remain living in the individual. You can follow and track the cells from the bloodstream, that they're still detectable, you know, months later. So we're seeing that some of these patients with partial responses went on to complete responses over time. So I think right now across the U.S., and I can't really speak for, you know, what's happening in other countries, but except there's a huge excitement. I think the frontline practitioner does want to treat, but ultimately all of us take patients and they become the primary focus of our attention. Primary, you know, practitioners want to treat their patients. They believe their patients should be treated locally, and I 100% endorse patients should be in their community. They should be close to home. Currently, these therapies are being still limited to academic centers with or highly supported community research centers where patients have to travel to get the therapy. I think that our current community oncologists are sending them willingly, but they certainly want the therapies to be developed to a point they can do it locally. Again, I would endorse that is the goal of therapy. And so what we have to do is, in this case, we have a therapy that works. What we have to also have a therapy that has toxicity. And what we need, I think the developments and a lot of the focus is either to get a therapy that can we make it more efficacious? Can we get more benefit achieved? But also, can we deliver these therapies safely, or even more safely, by finding ways that we can actually ameliorate some of the unique toxicities that are associated with these cell therapies?